So I started out my research career in Australia at the Garvan Institute in the cancer program. There I was head of a cancer genetics group, and we did start a lot of our work in prostate cancer actually here at the Garvan. I then subsequently left the Garvan to pursue two very large projects. Both of the projects were driven at that stage by technology, and that was technology that had really come to the fore in about 2005. And Australia was a little bit behind the US in adopting this technology, mainly because of the great expense involved in using the technology, and that was next generation sequencing. So I left the Garvan to be able to pursue these two projects, the first one being sequencing the human genome of the most ancient human lineages, being the Kusan Bushman people of the Kalahari Desert in southern Africa, as well as sequencing the first named African genome, being that of the Archbishop Desmond Tutu. The second project we pursued at the time was the sequencing of the tumour and the genome of the Tasmanian devil. This was a critical project which was needed for Tasmanian devil research because of the fact that the facial cancer is still spreading so fast, yet we didn't have a genome for scientists to work from. So we sequenced the Tasmanian devil genome and the tumour, did what we call a de novo assembly, which is piecing all the bits of DNA together without using a reference. And both of these projects required at the time very expensive technology, the 454 next generation sequencing technology, which was not only fairly expensive at that time with regards to reagent costs, but also the requirements as far as the laboratory space. Subsequently to doing those two projects, I was recruited to the J. Craig Venter Institute in San Diego, where we continued this research looking at human genome diversity and the extent of the diversity of the human genome. What we really want to understand from that project is what is this extent of diversity in the human genome that actually means being a healthy human, basically creating a reference for all research on medical disease. We still really to this day do not understand what it is to really be normal and what it is to be healthy. So for that reason, our researchers still continue to focus on the most ancient of our human lineages, being the Kusan people that live in the Kalahari Desert of southern Africa. For two reasons, not only because they carry the most genetic diversity within their genomes of all modern humans that are currently around, or contemporary modern humans, but also because they still live as close to possible to a hunter-gatherer lifestyle as we all lived over 10,000 years ago before the advent of agriculture. So they play a critical role in our understanding not only of ourselves and how modern humans have evolved, but also, and very importantly, they give us an understanding of a baseline to study human disease. So I've come back to the Garvan Institute, back to Sydney, back to Australia, and taken up the Petrie Chair of Prostate Cancer Research here at the Kinghorn Cancer Centre. This position is a joint position with the University of Sydney, and we have established a new laboratory in the cancer division, which is called the Laboratory of Human Comparative and Prostate Cancer Genomics. So we are utilizing the tools that we've learned over the years in genomics and in understanding the human genome to further our understanding in the human genome itself, as well as importantly to understand the consequence of human genomic diversity in driving prostate cancer, and in particular lethal prostate cancer. So as part of this exciting new adventure of being back at the Garvan, and as part of the Petrie Chair Foundation position, we decided as a team to acquire a new technology The technology is from a company called Bionanogenomics based in San Diego. And the technology we'll be implementing at the Garvan is called the IRIS system. Now, what is unique about the system, we know that there are many next generation sequencing technologies out there, and Garvan certainly has a large number of these technologies already in use. The big question is what makes this technology different? Well, this technology, as I see it, is really to answer what we call the missing genome, the parts of the human genome that we at this moment, using the current next generation sequencing technology, cannot answer, and cannot answer to the fullest extent. And what this is the current technologies that we use are very, very good at picking up single base changes, small changes in the human genome. What they're not very good at doing is picking up the very large changes, what we call structural variants. These may be large insertions, large deletions, translocations from one chromosome to another. And we know that these are very, very important in driving human disease, and in particular, it seems to be a significant driver in prostate cancer. How we know this is in our sequencing of many human genomes, and especially the most diverse genomes of modern humans, we have found that repeatedly, over and over, even if we sequence the same genome over, we do not necessarily pick up the same structural variants. We have a problem in repeatedly identifying them, as well as picking up what we believe to be a small percentage of what really is out there. So it is an area that still requires a lot of work. So we're extremely excited to have this technology. It's a technology that it's very early phase. We are the 6th instrument in the world, and we're the first for Australia. The reason why we're the first for Australia is there is going to be a lot of work on the team's behalf to get the technology working without the infrastructure and support that they have, for example, in San Diego. So we're going to have to be working across a time zone, but it's certainly extremely exciting for us, for the team, for the division, and for the Institute to be at the forefront of the next phase of genomics.